I'm Jacob Henson and I chose prompt 5.2, and today we'll be talking about Francis Crick's adapter hypothesis and how tRNA functions. I'd like to start out by laying a basis for the way tRNA works into translation of DNA. So I'd like to make an analogy to oversimplify the whole process. I consider myself a donut connoisseur, and we all know that Krispy Kreme has the best donuts around. And in a way, translation is like a Krispy Kreme. The ribosome is the site at which translation occurs. So the store is going to be like our ribosome. Then we have our mRNA, also known as messenger RNA, which will be like our conveyor belt that the donuts will eventually ride on throughout the process. Next, we have our tRNA, which stands for transfer RNA, which is the donut being placed on the conveyor belt. And tRNA molecules are actually placing an anticodon onto the codon that has been provided by the mRNA. And I'll show that in the diagram coming up. And a donut wouldn't be complete without icing. And it takes all these steps to build up to the icing of the donut or the cream being inserted into the donut. So this may be a stretch, but the icing slash cream slash sprinkles is analogous to an amino acid. Each type of donut, which has been determined by the codon slash anticodon relationship, is going to dictate the outcome of the amino acid. Finally, we have the production of a polypeptide chain, which is made up of our amino acids that we have coded for. I hope that this analogy has helped explain the parts associated with tRNA. So to explain the whole process, we have the ribosomal unit, which is serving as the site of translation. And inside of that is tRNA molecules, which are attaching and anticoding to the mRNA as it funnels to the subunit of the ribosome. As this happens, amino acids bind and keep binding until a protein chain is formed or whenever a stop codon enters the unit. Francis Crick proposed the idea of the adapter hypothesis, which basically states that small RNA molecules would be adapters that would receive specific amino acids. This hypothesis is heavily supported because the tRNA is ultimately making the placement of amino acids and the synthesis of proteins possible. The mRNA lays a template and tRNA takes up the rest of the work, placing an anticodon and making it possible for amino acids to bind. The name is fairly relevant because the whole process of tRNA is adapting that particular codon by adding an anticodon and allowing a highly specific amino acid to bind. If we were to talk about a particular alteration that would prevent tRNA from acting as an adapter, we could consider something like a mutation in which a change in the universal genetic code would prevent a proper adaptation to the codon from occurring. This would change cellular makeup. It wouldn't necessarily prohibit replication, but it would change cell differentiation and other cellular processes. The mutation would prevent the cell from meeting the needs of the organism.